We'll get started. First of all, welcome everybody for attending today's webinar. My name is Marie Zhang, and I'm a co-founder and currently the Chief Operating Officer at MicroStem. For the past few years at MicroStem, we have been developing technologies and products to enable primary cell culturing by recreating microenvironment in vitro. Let's spend a few minutes talk about microenvironment. As many of you know that microenvironment is very important for proper cell function and behavior, and it primarily consists of extracellular matrix proteins, signal molecules, and growth factors, along with other supporting cells. As you can see, the microenvironment is very complex and lots of factors. So in order to recreate such microenvironment in vitro, it's a very tedious and time and cost demanding process. And most of the time when they attempt to recreate such a microenvironment, they can only screen one factor at a time, which it's not truly mimic what's inside of your body. As we all know, there's lots of factors that work basically in concert together to drive cell behavior and function. So the result is, and to culture, especially to culture primary cells, it's been always very difficult just because the in vitro is not truly recapitulate what's inside of your body. So at the MicroStem, what we try to do is miniaturize this platform and the process. The way we try to tackle this problem by creating cell array. So what we do is we take a glass slice, and it's just the standard microscope glass slice, and we coat it with a hydrogel. Now this is another factor that many people kind of overlook. Inside your body, the cell does not really sit on a very hard surface. Instead, they sit in the kind of soft cushioned microenvironment. So our system and try to recreate this situation as well. So there's a hydrogel on the top of glass slice, and then on top of the hydrogel, we print a variety of different molecules and factors to try to recreate such a microenvironment. And the spot size are tunable. It can be small and it can be big. It all depends on the cell type that you want to study. And the hydrogel rigidity is also tunable. You can make it very hard. You can make it very soft. It depends on the study you want to conduct. We have lots of experience to print extracellular matrix proteins, and we have some experience printing signal molecules and growth factors and glycans and biopolymer. We have a lot of experience printing antibodies, and recently we have moved into printing nucleotide, polynucleotides such as DNA, RNA, particularly RNAi. And then we also try to work with a small molecule as well. And I want to tell you a little bit about the characteristic of our platform. And the spot, first of all, the spot size are uniform on the surface, and the material, when they deposit, they're retained on the spot and they are fully functional. And there's no cross diffusion between spots, because the particular hydrogel surface, it not only serves as a cushion for the cell to sit on, they also provide a surface for the materials such as extracellular matrix protein to be attached to. Such interaction is not covalent; it's actually non-covalent, and that's one of the reasons the matrix are fully functional on the surface. And I mentioned already that the hydrogel also provides surface rigidity. I want to spend a few minutes talk about the benefit that this platform can provide for the cell biologists. First of all, it's a high throughput platform, as you can see, because the spots are very small, and you can print thousands and thousands of conditions on the surface, and you don't need a lot of material, so it's very fast and economical. And because it's a miniaturized platform, so you don't need a lot of cells to study. And each spot usually have about 20 to 200 cells on it. So this makes it very applicable to lots of clinical samples. And because it's printing technology, so instead of coding like in the well format, so the deposits are very consistent of these materials, therefore enable very consistent experiment-to-experiment results. And this platform is very easy to use when people think about looking at a high throughput, high content system. They immediately think about, you know, the robotic system and the plate washer and things like that. And in our system, you don't need any of that. You can conduct all these experiments just in a very regular cell culture laboratory setting. And you can use a variety of imaging systems to image the slides. So it's very easy to use. I'm going to walk you through the workflow about how this process is like. And first, you get your cells, and the cells can be from a variety of different sources. It can be fresh, isolated cells, cell lines, frozen cells. We have tested them all. And the amount of cells that you need, for us, the smallest amount of cells we have used for primary cell culturing is about 20,000 cells. So you can see you don't need a lot of cells to study. And after you put the cells on the chip, basically first you put the chip in this 4-chambered cell culture vessel in one of the well, and it fits very nice snugly with the slides. And then you just load the cells on top. And after that, you can conduct your experiment. So far, we have studied cell attachment, viability, a lot of toxicity study. Those more in the more drug discovery kind of area. And for stem cell, we study extensively for proliferation and differentiation. Some of our customers have studied the transfections, and they have discovered that matrix actually affect the transfection efficiency, especially for primary cells. So that's a very interesting area that our customer has been using. So after you conduct your assay, you can just do the standard fixing, use a PFA, and then staining just like on the regular plate format. And then you can analyze the image using a variety of different methods. We routinely use a fluorescent microscope and use ImageJ to analyze the image. And you can also use a rescanner, which give you a little more overall global kind of signal. You cannot dive into very high detailed studies, but it's kind of a quick and dirty kind of analysis. But recently we have seen some very high density scanners, and they are very good. They're almost like optical system now. Of course, our system works great with high content imaging systems such as a thermal, ceramics system, and G's, InCells, NPs, Opera. And we have studied a variety of primary cell type using this system. And today my talks are primarily focused on the stem cells, such as hematopoietic stem cell, cardiac progenitor cell, mesenchymal stem cell, of course, pluripotent stem cell. We also have applied them to a variety of primary cells, such as neurons, hepatocyte, cardiomyocyte, and oligodendrocyte. And then there's another very big area that primary cell that I haven't listed here is tumor cells. So we study extensively on primary tumor cell culturing and the cancer stem cell, which I will talk a little bit later in this webinar. Okay, so I'm going to start to talk, for the stem cell application, I'm going to start talk about the pluripotent stem cell. So as many of you know, pluripotent stem cell is a cell type that by two major characteristics. First of all, they can replicate itself, and the second, they have potential to differentiate into 3 germ layers, such as a mesoderm, endoderm, and ectoderm. So there's a lots of different, there's a lots of papers and literature out there to describe the process and how to differentiate pluripotent stem cell into the 3 germ layers. But from what we have seen and heard is that lots of these protocols are not very robust, and some are not reproducible. So it seems that there's a demand for standardization of this process. So we look at this problem. We're thinking, you know, I think the challenge is that most people, when they do differentiation, they're still culturing their cell on the matrigel, and as many of us are aware that, you know, matrigel is undefined and it's a mouse tumor cells, and so we believe by replacing matrigel with defined matrices, and we should be able to improve the consistency and reproducibility of this process. So that was the goal for us to start this study. So again, we leverage our platform and to use this cell array process to look for the optimum xeno-free defined matrices for 3 germ layer differentiation. And the way we approach this is we designed 96 different conditions on the chip, and they consist of 9 different collagens, different extracellular matrices, including collagens, fibronectin, laminin, vitronectin, tropoelastin. And then we print them individually, and we also print them in combination. So there are total 96 unique conditions. And each of these conditions are printed 9 times. So there are 9 replicates. So really give you a lot of statistical information and confidence about your results. The center to center of the spot distance is 500 micron, and the spot diameter is 400 micron. We picked the 400 micron spot size mostly because the size of stem cell, and also because we're thinking how when they differentiate, what kind of cell type they could become. So the 400 micron will enable us to have between 50 to about 200 cells per spots. The workflow is very similar to what I just described a little bit earlier. And first, we culture the iPS cells to 80 to 90% confluency in the 6-well pre-coated stem matrix dish. I want to tell you a little bit about the stem matrix. This is a matrix that also developed here at MicroStem that is again replacing matrigel for maintain pluripotency for long-term stem cell culturing. So it's a defined matrix, and it's expressed actually in E. coli. So it has a potential to become a GMP kind of production matrix for future cell therapy. And so we use this to maintain our iPS cell lines, and we can passage them for 40 passages, and we'll still maintain the pluripotency, all the markers. And these are cultured on the NutriStem media. We also, this is a defined media, and also recently there's another defined media come out, it's called the E8. We also worked with the E8 so far at passage, I think at 20, the EA still work pretty well with our system. And then we just passage the iPS cells using Accutase into single cell suspension, and then we seed the iPS cells onto the slides in the 4-well plate. And after 24 hours, we remove the stem cell media, and then we add the differentiation media. And then we'll just take out the slide each day, and then stain for marker expression. And then all the slides will analyze, image will take and analyze on the Salome cell inside high content system. So this, one thing we were really looking at is see how many, what the matrix enable the cells to attach, and will allow them to still stay attached, after the differentiation process. So for ectoderm, we need 4 days, and endoderm we need again another. We also need 4 days, but for mesoderm we only need 2 days for our system to have a maximum marker expression. Again, I should have mentioned we picked the marker ahead of time. For ectoderm, we're looking at the SOX1, endoderm we're looking at SOX17, and mesoderm we're looking at the proprietary. So, and after 4 days, and then we were looking at what matrix do support the cell's attachment, and in this, graphics would kind of illustrate, on our 96 slides, and what are the conditions that still allow the cell to attach, and then also support the differentiation. Well, some of the phenomena was very interesting. Some of the conditions that initially attached, and then after we switched to differentiated media, the cells all left. And that means that particular condition will only support the pluripotent stem cell attachment in the stem cell media. Once you switch to differentiation media, that particular matrix combination do not support the differentiated cells, so that's why the cells gradually leave. Now, there's another something was interesting, was kind of surprising to us, is that for a certain matrix where initially there's no cells attached, probably because they wouldn't support a pluripotent stem cell attachment, but later on after differentiation, we saw these cells. We saw cells on these conditions. We were a little bit puzzled what happened. So I think our explanation is, even after we wash the slide, after we remove the stem cell media and put the differentiation media on, and some of the conditions that do not support the differentiated cell, those cells leave the attachment surface, and then because we observe cell every day, so we'll move the plate in and out the incubator, so those cells got kind of moved around, and then later on they actually find another favorable matrix, and they will settle down, they will proliferate, and they will differentiate again. So that was kind of very interesting for us to observe in our system. And then we, because our goal is finding the best marker expression and have the most cells attached to particular condition, so we can generate such a diagram looking at what condition allow you to find the most attachment, also have the best expression. Now, sometimes in the endoderm case for the SOX17, we have a very ideal situation where, for this particular conditions, ECM condition, you have lots of cell attached, and also you have the maximum SOX17 expression. In some other cases, you kind of have to make your judgment call. Like for this mesoderm case, you have some conditions have lots of cell attached, but it didn't have very high proprietary expression. So then you have to find some compromise where you think what you can, what your endpoint is, and you can make a decision yourself. So I want to show you a little bit of what are the, what they look like on these spots. For some of the conditions, and they have lots of cells attached on the top panel, but they don't really have lots of cells to have differentiated into the desired germ layer. In the top panel case, and most all these only have about 10 to 20 of cells have a desired marker expression. Now, some conditions kind of in the middle, they have significant cell attachment, and they have about about 40 to 70 cells that have a desired marker expression. Now, some of the condition here, they really have a very good attachment, and as well as very complete, almost complete differentiation. The amount of marker expression is above 95% of the cells. So we took these top conditions, and we decided that we're going to develop a kit that can allow people to QC their pluripotent stem cell to look at the potentials of how these cells can differentiate into the 3 germ layers. We evolved this kit into a 96 format instead of stay with the chip format. We move into the Lexis 12 format so that it will be more familiar to many cell biologists. So, so this kit consists of the optimal matrix, which discovered from the screening that I have previously talked about, and then we supply the all the differentiation media in the completely formulated format, and then we also supply in this kit validated antibody, the SOX17 and proprietary. The protocols are very simple. On the first day, you see 25,000 cells per well on a 96 well plate, pre-coded with supply the optimal matrix for each germ layer. And then on the next day, you remove the stem cell media and you put the differentiation media into the well, and then you just wait for desired days, and then looking and then fix and stain for the markers. So that's for the pluripotent stem cell study, and I want to move towards some study we've done for the adult stem cells. And the first example is the murine cardiac progenitor cells, and I think along with the next, the mesenchyms. These 2 studies are targeted towards cell therapy for a cardiac repair. And as many of you know, when people have a heart attack, their heart muscle died, and then they have a very difficult time to recover. And lots of time the patient have very difficult to recover the first heart attack, and then when the second heart attack comes in, that just really there's an enormous damage to their health. So, and we want to approach this problem in 2 different ways. And first, we want to see if we can use patients' own cardiac progenitor cells to grow them outside the body, and then transplant it back. On the next case, for mesenchymal stem cells, and because the difficulty of obtaining patients' own cardiac progenitor cell without another source of looking at these is looking at using its own mesenchymal stem cell, which is coming from bone marrow. It's easy to obtain. So for the first case for cardiac progenitor cell, because I should emphasize a little bit, this is on the murine system because it's hard to get access to human cardiac progenitor cells. They tend to be very enriched in one parts of the heart near the aorta, and usually during the bypass surgery, and the surgeon will slice off a small section of a tissue near the aorta, and that part is very enriched in cardiac progenitor cells. But regardless, it's a very invasive type of process. So we want to use a mouse model to start to investigate such a possibility. So what we did is, and I should also mention that the CPC cells are very rare in the heart. It consists of less than 0.1% of the heart cells. So what we did is we used a c-kit magnetic beads. c-kit is the stem cell marker, CPC stem cell marker, and then basically isolated these CPC cells. We usually take 5 mouse pups, and with 5, we can only get about 20,000 cells. And then we'll see on this particular custom designed screen, and with the 44 unique conditions, and we're able to identify a couple of them, then these CPC cells will attach and they will proliferate, which is demonstrated on this diagram. And then we also notice that for some of the conditions, although there's initial attachment, but the condition actually do not support the cell continue to grow. So after a few days, they actually die. But some others do support the CPC cell continue to grow on that particular matrix. Another thing what we notice is even in the same media, we notice some of the the ECM conditions actually support the stem cell characteristic, and some others actually already stimulate the CP cells to differentiate into cardiomyocyte, which is indicated in these two pictures. And the top is on one type of ECM. You can see all the stem cell marker c-KIT is very well expressed. But on the bottom, you can see the actin, which is cardiomyocyte marker, start to express. So for this condition, the cells already go into the cardiomyocyte differentiation pathway. For mesenchymal stem cell, as I just mentioned, it's easier than the CP cells to obtain in terms of the future cell therapy process. And mesenchymal stem cell is known to have the ability to differentiate in multiple cell lineage, including osteoblast, adipocyte, and chondrocyte. And the cardiomyocyte is probably the least studied. The other three cell types are very well studied. And we want to see, is there a way that we can leverage our screening platform to find the matrix, define the matrix to enable mesenchymal stem cells differentiation into pre-cardiomyocyte for transplantation. That's what the goal for this study. What we noticed is that after stimulation with azacitidine, this is a compound that's known to stimulate a mesenchymal stem cell towards a cardiomyocyte lineage. And we noticed on some of the conditions, the cells have a better NKX 2.5 expression, which is early cardiomyocyte marker, while others, although they do look morphologically, they seem to have changed, but the marker expression is not nearly as strong as some of these conditions. So this kind of gave us a very early clues of what ECMs that we can use in junction with other factors to potentially differentiate mesenchymal stem cells into cardiomyocytes, so that in the future we have an option for cardiac cells repair and to help the heart attack patient to regenerate the cardiac cells. I'm going to move a little bit now to hematopoietic stem cells. Again, hematopoietic stem cells is a blood stem cells, and they mostly are CD34 positive multipotent stem cells, and they can arise into all the blood cell type in the blood. And lots of people, there's a strong demand of being able to expand the hematopoietic stem cell outside the body, but so far has been shown that actually quite difficult, because every time when you try to expand, they differentiate, and we want to see is there a way that we can leverage our platform to find a way to allow the hematopoietic stem cell to expand in vitro. And there's a lot of people want to using these hematopoietic stem cells to looking for potential drugs, but then it becomes very problematic because they are supposed to be non-adherent cell type. So for high throughput cell image assay, the non-adherent cell types are quite difficult to screen. And we think, leverage our platform, we can find a matrix combination to allow them to sit down like a hidden cell type. And it kind of makes sense if you really think about hematopoietic stem cell inside of the bone marrow. They are not really floating. When they are proliferating, they actually, it's the bone marrow. They are in the microenvironment. They actually attach to something. They are not floating inside in the blood. So we worked with someone at the UCSF, and we also did conduct a study internally to design a special chip that have a lot of factors that we think can contribute to hematopoietic stem cell proliferation without differentiation. and it works actually surprisingly well. Even with the first try, we obtained some CD34 cells from a collaborator, and then simultaneously one of our customer also was working on the study, so she worked, she used the chip and also put her hematopoietic stem cells on it. These actually are from cord blood. And then we came to the same conclusion that we were able to identify several optimum matrices combinations to allow the HSC to attach and proliferate. And in our internal study, we went one step further. We actually stained them with CD34. In this panel, you can see for one of the conditions that although the cell seems attached and grow, but they lost all the CD34 marker expression, but on the other, they still maintain the marker expression, which we're pretty excited about because that's just really the very beginning for us to move forward to and develop a system to allow the hematopoietic stem cell to expand in vitro without differentiation. And this platform is also a adherent cell type of platform, which it really opened the door for many other studies, especially in the high throughput, high content type of setting. I'm going to move a little bit on something a little bit different for the cancer stem cell. As I mentioned earlier, we are very big focus in the company in the study tumor cells, cultured primary tumor cells, and because today's topic is cancer stem cell, it's the stem cells, so I thought I should talk a little bit about the cancer stem cells, and as a lot of us know that the cancer stem cells is it's a cell type that a very small population in all the tumor cells, and they are very evasive to chemotherapy and almost other therapies as well. And in primary stay dormant until they find an environment, then they can proliferate again. That's what caused the tumor. People think they cause tumor recurrence and metastasis. So it's a lot of interest that we should find a therapy to kill the cancer stem cell, which basically you not just merely treat the cancer, you basically cure the cancer. I think that's the word lots of people would like to use at some point in the future. So and then we also, because given our interest in this area, we were able to conduct one particular study. We obtained a lung resection sample from non-small cell carcinoma patient, cancer patient, and we use CD133 beads to isolate these subcell type. And then we again put these cells onto our chip and see if we can find the initial condition that promote the cell attachment. And the most importantly, they have to maintain the cancer stem cell marker expression, because they're from literature. Again, with these stem cells is once they attach, they differentiate. So we want to see if we can find a matrix that will allow them to attach but still maintain the cancer marker expression. So the preliminary data was pretty encouraging that we're able to identify actually multiple conditions. This is one of them that have a very good, still after 5 days, we still have a very good CD133 marker expression. So we continue to move forward in this study, and hopefully we will be able to generate a system to help people to study cancer stem cells and to help researchers to discover therapeutic treatment for cancer stem cells. Okay, I think that just concludes most of my presentation today. I hope after this talk and you agree with me that the matrix is a very important cellular function behavior, and we really believe our matrix array technology will allow people to do a very rapid screening and then to understand how the matrix influences stem cell growth and differentiation. And I want to spend the last few minutes talk a little bit about a couple of products we have, and the technology is pretty much embedded in this MicroMatrix product line. And this is basically a screening product that allow you to screen a variety of different ECM and combinations. Right now we have MicroMatrix 36, that's a 36 condition. The one I will just talk about, the 96, is coming up in the near future. That's more high, what I call a high density chip. For the 36, there are 36 unique conditions, and these you don't really need a high content imaging system. You can image them just in the regular fluorescent microscope and process them in the ImageJ and other imaging process software as well. And then we have a special chip specific design for neurons, because neurons require very unique matrix for its attachment and growth. And we do lots of custom design already. We have a customer who use our product first, and then they have some other matrix or growth factors they're interested in to study, and then we'll work with them, design a custom array, and we'll print them for them. So so that's something we are very interested in doing as well. The chamber matrix is kind of a hybrid between the slides and a plate. This is a 96 well spacing, and on the bottom still is a slide, but we put a chamber on top. There's 16 wells on this, and then we just print a variety of different matrices in the bottom. So if lots of time our customers start with this and they find a couple of optimal conditions to further study, and we'll custom build these product for them, and then they will use a different media, they examine different compounds, and they do assays on these kind of chamber matrix format. And we supply all the most of the ECM that printed on our chip, the collagen 1, 4, vitronactin, the fibronactin, and tropoelastin coming up as well. For the stem cell product, as I mentioned already, the stem matrix is for a long-term stem cell culturing while maintaining pluripotency. The C-stem is a product allow you to very rapidly looking at stem cell pluripotent stem cell markers such as Octa4, NANOP, and others. And the most recently, we launched the D-stem kit, which I spent some time to talk about today. And this is a kit allow you to very rapidly and in parallel to look at stem cells differentiation potential to 3 germinators. It's a very simple, streamlined protocol, very easy to use. For the neuron product, we have the MicroMatrix 36 neuron chip, which allow you to screen for the optimum matrix for particular neuron type that you study. And then because we did a lot of internal studies, we were able to actually have a matrix combination, which is actually a synthetic matrix combination to allow lots of neuronal cell to attach and grow. So these will be coming out as a neuromatrix pre-coded plates, and those should be available in the next couple of weeks. So I think that's what I have today, and if you have any question in the future, feel free to contact me. My contact information is on this slide, and I look forward to hear from you.
Okay, thanks. Yes,
Thank you.
Thanks a lot, and I will switch off the webinar.